So 1110. The idea of a signal transduction pathway is literally the process that a cell goes through to receive a message and then do something with that message. So signal transduction pathways usually involve this kind of set of steps. So in general, you should be able to explain what's happening in this picture or describe it. So you have to have a signaling molecule or a signaling cell that releases a molecule, and then that molecule is received usually by something on the outside of the cell. So that's called the receptor protein. So once that receives the signal, it causes some change inside the cell and starts the signal transduction pathway. So usually that means that it turns on a protein on the inside, and that protein turns on another protein, and that protein turns on another protein. And then at the end of that pathway, you end up with a transcription factor that's activated. So we talked about those yesterday. Transcription factors are needed to help transcription take place in eukaryotes. If those transcription factors aren't active and aren't present, then transcription of certain genes won't happen. So the purpose of a signal pathway is to turn on expression of a certain gene. So when you get the transcription factor activated, that means that transcription will happen on a particular gene in the DNA. So you get a specific sequence of mRNA, and then therefore you get a specific protein after translation. Okay, that's the point of a signal transduction pathway. So most of the time, the protein on the outside, again, has to be the thing receiving the signal. So most signals, most gene expression is turned on by something exterior to the cell. So there's a lot of places in this pathway where something can go wrong. We're going to talk about some of those. If you've read the chapter already, then you'll have seen some of the ways that mutations affect these pathways. But depending on what gets mutated or what part of the pathway is changed, you can cause the cell to make more protein than it's supposed to. You could prevent the cell from making protein that it's supposed to. Those could cause more cell growth or less cell growth than normal. And those things can help lead to cancer. Okay, so that's kind of the purpose of starting off with signal transduction pathways in the middle of the chapter. So in general, again, from here, you should know the steps for the signal transduction pathway. Cell signaling. So this 11.11 talks about the idea that cell signaling is something that's almost universal. So a lot of organisms exhibit this signaling or talking to each other, essentially. The example that's here is yeast having sex. So yeast cells, in order to mate, remember when cells that are simpler do that, we usually call it conjugation. So for cells to mate, yeast cells to mate, one cell that's an A type releases the A factor, that's the signal. And then another cell that's the other type of yeast releases its own signal. So based on the signals that are around a yeast cell, it can find a cell that it can mate with. Okay, this is like mating calls or courting rituals that some animals have, same kind of thing. It keeps a male from trying to interact with the wrong gender. Okay, so with the yeast cell, again, they receive the signals and then they grow towards each other. So in response to the factor that's in the environment, they go, oh, okay, I need to go that direction. So they grow or they move in that direction, and then you get a yeast cell that's mated. Okay. This is, again, fairly universal, so scientists use this as an evidence for evolution because it is universal, just like transcription and translation are universal. Okay, go for it.
How do the yeast multiply if they demultiply?
So that's a good question. If I remember correctly, once the cell has mated, then it duplicates the DNA. So now the DNA has both types, and it can divide. So you have a cell that still has both types of DNA, but it's duplicated. Later on in the process, it splits again. So you'll get a cell that has one half versus the other half. So yeast have multiple forms. There are other organisms that do that too, where one form has, like, for example, 2 copies of the chromosome, and the other form only has 1 copy. That makes sense? So you can kind of go back and forth. Some organisms that do that, there are a lot of plants that do that. I want to say that jellyfish do that as well. So the jellyfish have 2 different life stages. There's the stage you're used to seeing, and then there's a stage that's actually sedentary and it doesn't move around and it's stuck like to the bottom of the ocean. So those 2 forms kind of alternate how much DNA they have. So Okay, good question. Moving on, talking about cloning plants and animals. Realistically, this says cloning plants and animals. This section's really just about plants. The next section is about cloning animals. But we do clone plants all the time. Ever had a seedless watermelon? That's a cloned plant. Because if something is seedless, it can't produce seeds that you can plant. So the only way to get that type of a plant is to clone it. Now, this type of cloning is not cloning that we think of like what you would see in a science fiction show per se, because it's not nearly as complicated. Cloning a plant is not hard. So the example here, as far as cloning goes, that they actually show you is a carrot cell. So if you take a piece of a carrot and you put it in the right culture, from one single carrot cell, you can grow a whole nother carrot. Okay? So plants do this all the time. And if you know anybody who's ever given somebody else pieces of their plant to plant in their own garden, that's plant cloning. We just don't usually think of that as cloning. Okay? So if you do that, if I have a cactus, so cactuses can do this, or some succulents can do this. Some succulents look like they have leaves almost. So if you take a leaf off and you can get that leaf to grow roots, you can plant it, and then you have a new succulent plant. The original plant and the new one have the exact same DNA, so it's a clone. It's a type of asexual reproduction, but it's not cloning the way we like think of growing a test tube baby. So it's a little different, okay? But it's still, again, genetically identical, okay? So terminology that you should be aware of is this term. It's totipotent or totipotent. I've heard it said both ways. And this literally means that a cell has the ability to become different types of tissue. Okay, so it is able to turn into the root of the carrot. It can turn into the leaf of the carrot. It could turn into the actual part of the carrot that you eat. That's cell differentiation, becoming different types of tissues or different cell types. So if something is totipotent, it can be turned into anything else. Okay, there are the one other thing from here, they mentioned a take-home lesson. This is from the whole chapter, not just this section, but we've come back to the idea that genes only turn on a really small percentage, cells only turn on a really small percentage of their genes. That's a big lesson from this chapter, because again, you don't want one cell to try to do all the things. You don't want to express all the protein. You just need protein for that one cell to be a specific cell type. So when we're looking at differentiation, that's the cell only turning on a certain set of genes so that it becomes a root cell versus a carrot cell versus a leaf cell. Okay? So if you can take a single cell and then, under the right conditions, make it turn into something else or start differentiating differently, then it's totipotent. Okay, so at the end they talk about, okay, well, we can clone plants. We've got this down pretty well. Can we clone animals? Great question. So to give some examples in here, they talk about an indication that animals should be able to be cloned is the idea that if you get cut, you heal yourself. Okay, yeah, go.
What about starfish?
Okay, yeah, starfish are another good example. Their other example here are salamanders, I think. So if a salamander loses a leg, then it can regrow the leg. Okay, starfish do the same thing. If you cut off, I think you still have to have a piece of the center, but some of the cells in the center are totipotent, so they can regrow the whole starfish. Other examples, pineapple does the same thing. So if you take a pineapple from the store and you slice it from the top down in the middle, you can slice it 11 different ways and grow 11 pineapple plants if you want to. Granted, they won't survive here because our climate's not the right climate for them to grow in, but you could start growing 11 pineapple plants. Okay, so yeah, starfish are a good example. Sponges are another example. They name a couple of others in here. Okay, so then animals. Ready? Cloning animals. Usually when we clone animals, we use a process called nuclear transplantation. And you should be able to just generally describe that. So it's written out here. This is a picture or diagram of what it looks like. So nuclear transplantation is taking the nucleus from an adult cell, So this is an adult somatic cell or a body cell, and inserting that into an egg cell that you've taken the partial nucleus out of. Okay, so you have the nucleus from an egg cell that you remove. You take the somatic cell or the nucleus from a somatic cell and insert that in. So even in this process, a lot of what you're trying to create as a viable zygote or a viable cell to then grow from, they don't all survive. So this process destroys a lot of cells, okay, just so you know. And then if you put that cell, once it's successful, if you put that in the right environment, you can get it to grow into an embryo. Or the early term for an embryo is a blastocyst, which literally means ball of cells. So it's a little blob, pretty much. From the blastocyst, though, then you implant that into a mom, into a female, and then that should develop like a normal embryo. So this is pretty similar to how in vitro fertilization works. So if you know anybody who's gone through fertility treatments and tried IVF, this would be what it looks like. So in order for in vitro fertilization to work, you get usually several embryos that are in this stage. So you have several fertilized egg cells, and then those are usually left frozen as you try to put one or two into the person who wants to get pregnant at the same time. And if that egg implants, then they're pregnant. Okay, so that's kind of what that process looks like. Have you guys heard of Dolly, the sheep? Dolly the sheep is one of the first things, actually it might be the very first thing that was cloned in 1996, so that was a while ago. Okay, so Dolly was one of the first successful clones that was born, and they describe kind of what that looks like here. But Dolly demonstrated for scientists that yes, we can clone animals. So since Dolly, scientists have cloned a bunch of different things: cats, mice, cows, mules, pigs, rabbits, dogs, monkeys. Okay, so we've cloned a lot of different animals. And in doing that, scientists have done that first to see if they can, but also to try to test different things. So improving techniques for cloning. They're trying to do things like seeing if they can improve agriculture, or specifically breeding cattle or breeding organisms for different purposes. But in that, we've learned that there are some problems with cloning. So underneath where my sticky note is and up over here, it talks about pros and cons of cloning. So some of the pros are things like the San Diego Zoo in California currently has, I think it's more than 900 different embryos frozen, so that hopefully one day they can form clones from them and have those organisms be around again. So they have DNA for some things that are endangered. They have DNA for some things that are already extinct. So they're kind of hoping that, well, maybe one day we can develop to the point where we can bring those species back. Okay, most people view that as a positive thing. Okay, some cons though are the health of clones seems to be worse than the original animal, or an animal that's just produced through normal natural means. So clones tend to develop cancer more often. They have a lot of other health problems, and only about, let's see if I can find the number. I think it's about 10%, somewhere in this section. Yep. Only less than 10% of embryos develop normally. So that means that even if you have a clone, the embryos formed correctly, so this step here, once it is implanted, you might still have a miscarriage because the embryo might not develop the right way, and the pregnancy might get ended early. So that's really low success rates. So if that's our success rate with animals, then people go, well, can we really clone humans? Is that ethical? Is that right? Okay. The book doesn't address whether it's right or not. They just say there are ethical concerns with, and leave it at that. So there's as many opinions on whether or not that's right as there are opinions about pretty much anything else, politics included. So you can imagine some people are really passionate about it. Some people have really strong opinions. So there are just differences with that. This section talks about a different type of cloning. This is therapeutic cloning. So if you remember when we watched the designer DNA video, there was a section that talked about therapeutic genes or using genes as therapy. The idea of therapeutic cloning is when you try to clone for the purpose of healing a disease. So we're not trying to make a whole new organism or a whole baby, but we are trying to maybe grow a new heart or help cure someone's lack of a specific protein. So therapeutic cloning involves the use of stem cells. And there's 3 general types of stem cells. There's embryonic stem cells, there are adult stem cells, and there's also umbilical cord stem cells. So fun fact, in case you didn't know, the name for your belly button is called the umbilicus. So that's why we call it an umbilical cord. There you go, fun anatomy term, okay? So with embryonic stem cells, this requires that you take the cells from the blastocyst. So it means that you have to have an embryo that then you pull cells out of. So you destroy what's left of the embryo to get embryonic stem cells. But from embryonic stem cells, we know that we can put them into different cultures or different mediums and cause them to grow into different types of things, okay? With adult stem cells, they are not as totipotent, so they don't differentiate into as many different things. So adult stem cells in bone marrow will only become blood cells. We haven't figured out how to reverse turning them on into nerve cells or heart muscle cells. And umbilical cord stem cell research at this point is the same way. There hasn't been a lot of success with that. So embryonic stem cells are something that a lot of people want to use for research, but again, because it requires destroying an embryo, some people feel very strongly about that. So there's a wide range of opinions, just like do you clone animals or not? Wide range of opinions, okay? We'll talk about the cancer stuff, which is the last few sections.